Hello everyone and welcome to the NPTEL online course in pharmacognosy and phytochemistry. In the previous session, we learned about what are tannins and what are different classes of tannins. Today we delve little deeper into that. We will learn the different sources of tannins and the methods of evaluation of tannins in different substances present in nature. In nature, tannins are present in green tea. When you say green tea, it's the light green color concoction that you produce out of tea leaves, which contains epigallocatechin gallate and the very tarty, very astringent amla. They contain low molecular weight pseudotannins such as gallic acid and epigallocatechin gallate. Apart from that, true tannins, which are high molecular weight tannins, are present in form of hydrolyzable and condensed tannins. The hydrolyzable tannins, gallotannins, are present in nut galls. They are present in different fruits and berries such as strawberries, raspberries, mostly in form of pentagalloyl glucose as a smallest unit. They are in form of elagitannins in pomegranate peels, in form of punicalagin, and in Harday fruits they are present as chebulagic acid. When you go to non-hydrolyzable tannins, you have condensed tannins, the flavonol type, which are present in pale and black catechu, and fluorotannins, which are present in sea kelp. Apart from that, complex tannins are found to occur in Camellia japonica in form of Camellia tannins A and B. So these are some sources of tannins. You will find tannins occurring in numerous plants as they are protective molecules. So how do we evaluate them? The first step to evaluate tannins is to extract them in a suitable solvent and then subject them to different chemical methods of evaluation. The extraction of tannins is done in a solvents which are polar in nature. You can extract tannins with water, ethanol, acetone or mixture of them. That is, you could use 50% water, 50% ethanol or 50% acetone and 50% water as a mixture to extract tannins. The solubility of tannins in these solvents depend upon their molecular weight. The higher the molecular weight, more is the resistance to its solubility in different solvents. When you take tannins, it is best to take the drug in its fresh form. The reason being, I just illustrate it with the example of tea. When you take a fresh tea leaf, it contains tannins in a very low molecular weight form. But as you ferment, as you roast the coffee beans, the tannins polymerize in themselves and are converted into high molecular weight polymers. These high molecular weight polymers are difficult to extract. Hence, in order to assess the tannins of a drug substance, it is best to extract them or keep the sample frozen till you need it for the chemical evaluation. Now, when you take a sample, sometimes there are certain samples which are extensively lipophilic. That means they contain oleaginous ingredients. This includes your oils, butters or waxes. When you have oils, butters or waxes present in a plant, they don't allow solvents such as water to imbibe deeply into the tissue. And that is the reason when you want to evaluate such drugs for their tannin content, the first step is to remove the waxy contents. The waxy contents or the lipid contents or oils can be removed using organic solvents such as petroleum ether or hexane. They will dissolve these lipophilic compounds and now your tissue is free of them. Once the tissue is free of all the lipid compounds, the water penetrates much easily and your compounds are extracted completely. The other problem is when your plant product is a leaf or a stem which contains pigments. Now, these pigments can be something like your chlorophyll. Chlorophyll often interferes with color evaluation because inherently it is green in color. To get rid of chlorophyll, it is advised that you extract the sample with chloroform or organic solvents in which your chlorophyll has a good solubility. Chloroform will dissolve the chlorophyll, and then once the pigments are removed, the tannins can be easily assessed using colorimetric test. Now, once you get the tannin extract, you can further purify them by subjecting it to different chromatographic separation techniques. Now, let's see how do we assess them. Once you have your extract of tannins ready, you can apply few general tests to see whether they contain tannins or not. The general tests for true tannins include Gold-Beater's test, which uses a ox skin, gelatin test, which uses a gelatin solution, lead acetate test, and a phenazone test. This test will tell you whether the two tannins are present in solution or not. Let's see them one by one. So for the Gold-Beater skin test is actually a test where you convert your animal skin into leather. Now this process is mimicking your tanning process. So what you take in for a tan or what you take in for an animal skin is actually an intestine of an ox soaked in a hydrochloric acid solution. So we'll take a membrane or intestinal membrane of an ox which is untanned and unused, and then we will soak it in acid to get rid of the initial impurities and to make the skin more amenable to tanning. Once that is done, the acid is washed clearly, and then what you can do is you add a tannin solution. This can be done by just taking a small container containing your tannin solution, and you can just keep your skin or the ox skin immersed in it for a period of about 5 minutes. The skin slowly imbibes all the tannins. The tannins will react with the proteins present on the surface of skin, and slowly the complex formation starts. Once the complex formation is done, after a period of 5 minutes, the tannin solution, after a period of 5 minutes, the tannin solution is discarded. The ox skin is nicely washed with water and then dipped with a solution which is called as mordant. Mordant act as a fixative. So it will help intensify the color, convert the animal hide into a deep brown black color leather, and that you can easily assess by change in color of the or change in appearance of the ox skin. If that skin turns brown or black, we say that the solution contains tannins. So that's a positive Gold-Beater's test. Now if your solution contains pseudotannins or low molecular weight tannins, the intensity of color change is low or negligible. So this test is generally not indicative of pseudotannins, but is a test for presence of true tannins. The next test what you can do is also a test which combines tannins with proteins, and that is your gelatin test. In this gelatin test, we use gelatin as a source of protein because gelatin comes from your cartilage. That is the hydrolysis product of the cartilage. So gelatin is taken, prepared in a salt solution, saturated, and to that when you add your tannins, these tannins form complexes with gelatin and slowly precipitate. As you can see here, initially you have a very clear tannin solution, but once this tannin solution is added to a test tube containing gelatin with salt, you can see a deep color precipitate. This is what is called as tanning. So a tanned blackish brownish color complex has been formed, and if you allow it to settle over a period of time, it will settle down, giving you a precipitate of the complex. The next test what we can do is a lead acetate test. In this test, actually the ability of tannins to form complex with lead. Now tannins have a ability to chelate metal ions. With lead they form lead tannate. So when my tannin solution I added to a lead acetate solution, a yellowish color or yellowish whitish precipitate of lead tannate is formed, which you can see here is indicated by a turbidity. This turbidity, again, if you allow to mature over a period of time, it will settle down. The presence of whitish to yellowish precipitate is indicative of tannins present in your solution. Another test which is indicative of tannins is a phenazone test. So if you take a tannin solution and mix it with a sodium acetate phosphate, boil it, filter it and then cool it. So after cooling, when you react it with 2% phenazone solution, you get a bulky precipitate. That is also indicative of presence of tannins in your solution. Now with that, let's move on to some specific tests. So specific tests for tannins include ferric chloride test. Ferric chloride test helps us distinguish between hydrolyzable and condensed tannins by the color formation. The second test is a potassium iodate test, which is more specific for gallotannins and gallic acid. Nitrous acid test, which is specific for elagitannins and ellagic acid. The vanillin hydrochloric acid test, which is given by flavanol derivatives, and the matchstick test, which is again given by condensed tannins, the flavanol derivatives. Now let's see them one by one. So the first test we analyze here is a ferric chloride test. Now why this is distinguishing test is, if you take a tannin solution, as you can see here, this is a tannin solution. Principally tannins are buff to brown color compounds, so your extract appears something like this. Now I have taken one extract which is rich in hydrolyzable tannin and one extract which is rich in condensed tannin. So what happens here is the extract containing condensed tannins, when I add a drop of ferric chloride to it, you will see a greenish coloration, whereas in case of hydrolyzable tannins, you will see here a bluish coloration being formed. So this test helps you distinguish between blue or green color complexes which are formed by reacting with hydrolyzable and condensed tannins respectively. So let's see what happens exactly out here. So in this process, you can see gallic acid as an example of a hydrolyzable tannins or a gallotannin. Now this gallic acid reacts and forms coordination complex. So you can see here, they form coordination complex with iron, and this chelate gives you a typical coloration. So you can see how the process of addition happens. This is a tube containing light or very dilute solution of tannins, and I've added a drop of ferric chloride. So at the interface, when the reaction is happening, you can see a blue color complex being formed. Similarly, you can even check for the presence of specific hydrolyzable tannins such as gallotannins and elagitannins. If we want to check for the presence of gallotannins, what we can do is take the solution containing gallotannins and react them with potassium iodate solution. If you get a presence of pink coloration, it is due to the presence of gallotannins. Now instead of gallotannins, if we have pseudotannins like an individual monomeric gallic acid molecule, in that case you will get an orange color. Now we can also check for the presence of elagitannins. Elagitannins form complexes with nitrous acid in an acidic pH, that is in presence of acetic acid, to give you a pink color. Now this test is obeyed both by elagitannins also and monomeric ellagic acid also. So both of them will initially form a pink color complex, and gradually and transiently the color of this complex will change initially to purple and then to blue color. So presence of a transient pink to bluish coloration is an indication of elagitannins being present. Now moving to condensed tannins. Condensed tannins, especially the flavanol type, react with ferric chloride to give you complex. Now this is similar to gallic acid, but you can see the nature of complex is slightly different. So here also chelation does happen, that is the coordination complexes, and you can see. Now because of a bulkiness, resonance of this kind of structures, instead of a blue coloration, you will get a green coloration being formed. So you can see here at the interface, as compared to your hydrolyzable tannins, condensed tannins give you a green color complex. Another test for condensed tannins, especially the flavonol type, is when you treat it with vanillin hydrochloric acid. Now vanillin in presence of acidic pH reacts with flavin, especially the meta-substituted flavin derivatives, to give you a reddish color complex. Now this is an electrophilic reaction where you form or where you get a vanillin adduct. Now this vanillin adduct is again not selective. So even if you have a plain flavin 3-ol derivative like a isolated catechin molecule in monomeric form, still it will give you a red color complex. So vanillin hydrochloric acid test is a test which is used to check flavin-ol derivatives, which gives you a red color complexes. Interestingly, this test is also used histochemically. So what you need to do is you can take your section, that is a transverse section. In that transverse section, just add a drop of your vanillin solution, that is alcoholic vanillin, let it dry off, and then you add a drop of concentrated hydrochloric acid. What will happen is wherever your condensed tannins are present in the section, that section will tend, or that area of section will tend to form a reddish zone. The presence of a reddish zone in your section indicates that your condensed tannins, especially the flavin 3-ol derivatives, are present there. Another test for tannins, especially the condensed flavin 3-ol derivatives, is your matchstick test. Now this is a very interesting test. If you see the matchstick comes from wood which is rich in lignin. So this lignin derivatives have cinnamaldehyde groups, and this cinnamaldehyde groups react with the catechin or flavin 3-ol derivatives. Now here is an example of a molecule of catechin. So what happens is when I dip my matchstick in a solution of catechin or a condensed tannins and just get it near to flame, it will start developing or reacting with the cinnamaldehyde functionality. As it reacts, if I give it a concentrated acid such as hydrochloric acid, just by dipping the matchstick in concentrated hydrochloric acid, slowly this molecule breaks down to give you what is called as fluoroglucinol. Now this fluoroglucinol has a tendency to react with the lignin to give you a purple color complexes. So lignins react with this free fluoroglucinol moiety and I get a presence of purple magenta shade. You can see here initially the matchstick is nice yellow in color, but after I dip it in catechu, you can see the color is brownish, and here the region where it is dipped in HCl and brought near to flame, you get a presence of purple to magenta color indicating the presence of condensed tannins. Now apart from that, we can even quantify tannins. The general methods for quantification of tannins are based on the reaction of tannins with proteins. So they might react with blood components to give you a hemolyzed blood method. They might react with proteins in bovine serum to give you a bovine serum albumin method. They might react with the proteins when present in animal hide to give you an animal hide method. So how do this work? So these three methods are basically complexes of tannins plus proteins. So just let's try to understand them. In hemolyzed blood method, what is done is blood is treated with tannin sample. The proteins in the blood will settle or form complexes with the tannins. The sample is centrifuged and the amount of deposit is quantified gravimetrically. Same is done for bovine serum albumin, but the method is reverse. So first we quantify how much bovine serum albumin is present. Then you add a tannin solution. Some amount of this bovine serum albumin will react with the tannins. They form precipitate. The tannin removes the amount of serum albumin equivalent to its moles, and that much amount of bovine serum albumin is now gone off the solution. Again, when we quantify the bovine serum albumin, the difference in weight of bovine serum albumin will tell me how much tannins are present. Same applies for animal hide powder. You can prepare a suspension of animal hide powder by boiling it, making it little hydrated, and then reacting it with tannin. The amount of precipitate will tell you how much tannin is present. The last method is a non-specific method. It is used for determination of polyphenols in general. So Folin CO-Cal2 reagent consists of phosphotungstic and phosphomolybdic acid. They form adducts, blue color complexes with tannins under basic pH. So this blue color complexes, intensity of this blue color complexes is directly proportional to the intensity of polyphenolic compounds being present. Now since tannins are also polyphenolic, we can use Folin CO-Cal2 method to quantify tannins using UV-Vis spectroscopic method. One more method for quantification and analysis is your thin layer chromatographic method. This method uses your silica as a stationary phase and organic solvents such as toluene, ethyl acetate, methanol, formic acid, a combined phase which we call it as mobile phase for running this. The principle is, in this case, your stationary phase is polar and your mobile phase is non-polar. So tannins, depending upon their affinity to the stationary versus mobile phase, will start running on the plate, and they run to a specific distance. We call it as a retention factor, RF. So if you can see here, we have a plant extract being applied, that is two different plant extracts, and here G stands for gallic acid. So if you see, gallic acid travels a certain distance and you can simultaneously see a corresponding band in the plant extract. What does this indicate? This indicates that my plant extract also contains gallic acid. TLC along with a scanner called as photo documentation can also be used to quantify the amount of tannins present based on their RF and intensity. Another methods which have been used for tannins are HPLC, size exclusion, and few of them also use GC, but those are beyond the scope of this. So thank you everyone for your patient listening. Here are few references just in case you wish to know more about this. Thank you.